White blood cells are classified according to the appearance of granules or lack of granules. For example, in the top right corner of this slide you see a basophil. A basophil is only one of three granulocytes. The other two are eosinophils and neutrophils. Why are they called granulocytes? Simply put, when stained, the cells appear as if they have grains of sand in their cytoplasm. It appears blotchy. It really isn't grains of sand. Rather, they are components in the cytoplasm that bind to the acid dye eosin, as in the case of the eosinophil, or the base dye methylene blue, as in the case of the basophil, or both dyes, as in the case of the neutrophil. Granulocytes are also called polymorphonuclear cells, which means mini-shaped nucleus. When one looks at the nucleus of these cells, it appears contorted, much like an animal balloon. Its appearance might trick you into thinking there are many nuclei in the cell when really there is only one. Next, how will you remember which white blood cells are granulocytes? Well, notice they all end with a suffix "phil." Basophil, eosinophil, neutrophil. So just remember that the granulocytes are, quote, filled with granules. Agranulocytes do not contain visible granules when stained, and the white blood cells in this category are the monocytes and lymphocytes. In the bottom right picture, you can see a monocyte with its U-shaped nucleus. One question I frequently get from many anatomy and physiology students is, why do the nuclei of white blood cells, and other cells of the body for that matter, always stain a purple color? A nucleus is packed with acids, nucleic acids. The dye, methylene blue, is a basic dye, and therefore the nucleic acids in the nucleus binds to the methylene blue, and the nucleus appears stained a bluish-purple color. The life of the granulocytes after release from the bone marrow is 4 to 8 hours circulating in the blood and another 4 to 5 days in the tissues where they are needed. In times of serious tissue infection, this lifespan is even more abbreviated to a few hours. Why? Because it is the granulocytes that proceed even more rapidly to the infected area, perform their function, and in this process they are themselves destroyed. I will elaborate on this inflammatory response later. Let's start our highlight of the white blood cell with the basophil. The basophil represents 1% of all circulating leukocytes. The granules within the basophil soak up the methylene blue dye and thus stain a dark purple or blue. Being a polymorphonuclear cell, the nucleus is bilobed and has an S-shaped appearance. Basophils migrate to injury sites and release the contents of their granules. The granules contain histamine and heparin among other components. Histamine is a vasodilator and increases capillary permeability. Collectively, this means the capillary will have more blood flow through it, and the capillary itself will be more, quote, leaky to allow more white blood cells to reach the tissue infection. Heparin is an anticoagulant, and this means that the blood will be less likely to clot and allows better movement of the white blood cells to the infection site. The basophil's function really augments the inflammation response initiated by the mast cells. I always remember B for basophils and for, quote, begin, as in beginning the inflammatory response. The next granulocyte I'd like to highlight is the eosinophil. This granulocyte absorbs the acid dye eosin, and the granules stain red as a result. The nucleus is bilobed and has the shape of an old-fashioned phone receiver. Like other granulocytes, this cell travels in the bloodstream and then enters the tissues, and its lifespan can be anywhere from minutes to days, depending on the severity of the infection it is fighting. How does an eosinophil fight infections? Well, it is up to speculation whether or not it phagocytizes antibody-coated material, but their main method of attack is through exocytosis of toxic compounds onto the surface of their target. Let's say the eosinophil encounters a helminthy worm in your digestive tract. The eosinophil will release hydrolytic enzymes from their granules, which are basically modified lysosomes, onto the surface of the parasitic worm. Alternatively, the granules can release reactive oxygen species like hydrogen peroxide, which will create holes in the parasitic worm's cell membranes, or they can also release a larvicidal polypeptide called major basic protein. Regardless of its method of attack, you should have this take-home message that eosinophils attach themselves to the parasites, particularly those in your GI tract, and then release hydrolytic substances and digest the parasite from the outside in. During the time of infection, the eosinophil numbers will increase. Also, eosinophils seem to play a role in preventing the excessive spread of local inflammatory factors. They also help in ending the inflammatory responses initiated by mast cells and basophils. Clearly, if the mast cells and basophils were left unchecked, then widespread histamine and heparin release would cause excessive vasodilation. This is what happens during anaphylactic shock. In a healthy individual, the eosinophil helps temper the mast cells and basophil activity by modulating the spread of inflammatory factors, referred to as cytokines, to maintain homeostasis during an allergic reaction. The last of the granulocytes that I'm going to describe are the neutrophils. Their cytoplasmic granules absorb both the acid eosin dye and the basic methylene blue. Therefore, their granules appear pale or neutral colored. The granules contain bactericidal compounds, and their polymorphonucleus is multilobed. These cells are the most abundant of the circulating white blood cells. This white blood cell is extremely mobile and usually the second white blood cell to arrive at an injury site. The macrophage, residing in the tissue, is clearly first, and this will be described later in this lecture. The neutrophils remain in the bloodstream for only a few hours, and then they migrate into the tissues to help fight infections and live for about 4 to 5 days. How do neutrophils fight infections? Well, they use a process called opsonization. When antibodies released by a mature B lymphocyte bind to a foreign particle or antigen, the antibody-antigen complex can then bind to surface receptors on the neutrophil. This basically immobilizes the antigen and affords the neutrophil time to phagocytize the foreign particle. In the movie The Blob, a big gelatinous monster slowly crept around and enveloped the victim internally, and then its victims were slowly digested. This movie basically helps describe how some white blood cells, such as neutrophils and macrophages, phagocytose foreign materials such as bacteria and other microbes. In this picture, I show you an aqua blue bacterium covered with antibodies. They are shaped as upside down, quote, Ys. When a bacterium is covered with antibodies, the antibody tail can dock to a cell surface receptor found on the neutrophil and macrophage. This is called opsonization, and the advantage to having antibodies that help dock a bacterium to this receptor is that the bacterium is now unable to move away as the macrophage or neutrophil wraps itself around the bacterium to engulf it. I have provided a link on this slide that will direct you to a movie that shows a macrophage engulfing a large glass rod. It truly is worth seeing. It is now time to address the other type of white blood cells called agranulocytes. Our first example is a monocyte, shown here. Monocytes are agranulocytes because when stained and looked at under a microscope, they lack granules in their cytoplasm. Monocytes have a U-shaped nucleus, and they stay in the bloodstream for about 24 hours. After this time, they enter the connective tissue all over the body and swell up to a much larger size to become a mature tissue macrophage. It takes approximately 8 hours for a monocyte to mature into a tissue macrophage. Macrophages are considered to be antigen-presenting cells. This means they help activate other white blood cells. Let me explain. Most invading organisms are first phagocytized and then partially digested by macrophages. The antigenic products are then liberated into the macrophage cytosol. The macrophage then displays part of the digested material on their cell surface and can pass these antigens onto other white blood cells such as lymphocytes through cell-to-cell contact. This process then leads to the activation of those lymphocytes that come into contact with a specific antigen that they can recognize. Lymphocytes include T and B cells. Perhaps you need another way to wrap your mind around this. I'm sure you have encountered children wearing messy shirts. Perhaps you could identify exactly what it was that they were eating. Maybe some ketchup from their hot dog, maybe some punch, or sticky cotton candy. Macrophages are very similar to these children. Whatever they have eaten, they display on their cell surface for other white blood cells to recognize in order to activate them, much like a mother or father would be activated into cleaning up after their messy child. Tissue macrophages reside everywhere in our body. In connective tissue and skin, they are called Langerhans cells, and in the liver, they are called Kupffer cells. Macrophages are our first line of defense. They phagocytize bacteria, break down the bacteria in lysosomes, display parts of the foreign material on their cell surface, and recruit and activate other white blood cells to come join the battle. Let's now go over again some of the important qualities of a macrophage. It is an agranulocyte and a very potent phagocytic cell. It can ingest many more foreign particles than a neutrophil, and it is your first line of defense against invading microbes, followed by neutrophils. The macrophage is considered to be a nonspecific immune cell. By that I mean it does not discriminate between the type of material it ingests. It can engulf any types of bacteria, other microbes, or even dead or dying body cells like red blood cells. It will wrap itself around foreign particles or cellular debris, internalize those particles, and then use lysosomes to break down the particulate matter. Lysosomes are cytoplasmic vesicles that are filled with hydrolytic enzymes. When the particle has been broken down by these enzymes, the macrophage will discharge the waste materials. Some of those particles will remain embedded in the cell membrane of the macrophage, so that the macrophage can act as an antigen-presenting cell and activate T cells and B cells into action. The other type of agranulocyte that I need to discuss is the lymphocyte. When you look at this picture, you'll notice that the lymphocyte has a very large, dark staining nucleus with a thin, pale halo of cytoplasm around it. Much like the other white blood cells already discussed, the lymphocytes travel in the bloodstream for a short time and then migrate into connective tissues and lymphatic organs where they fight foreign antigens. The two main classes of lymphocytes are T cells and B cells. T cells provide your cell-mediated immunity and fight infections like foreign microbes through cell-to-cell contact. B cells provide your humoral immunity, and they fight infections by producing antibodies against a specific antigen. Antibodies are immunoglobulin proteins found within the plasma of your blood and bind to specific antigens to neutralize them or destroy them. Antibodies will be discussed in more detail later on in this lecture. Leukocytosis refers to excessive numbers of white blood cells. Uncontrolled production of white blood cells can be caused by a cancerous mutation of a myelogenous or lymphogenous cell. What does that mean? Remember that the hemocytoblast is a multipotent stem cell that gives birth to myelogenous daughter cells such as neutrophils, eosinophils, monocytes, platelets, and basophils, or lymphogenous daughter cells such as T and B lymphocytes. When there is a mutation in the myelogenous or lymphogenous cell lines, this causes leukemia, which is characterized by greatly increased numbers of abnormal white blood cells. The lymphocytic leukemias are caused by cancerous production of lymphoid cells, usually beginning in the lymph node or other lymphocytic tissue, and then spreads to other tissues of the body. The other type of leukemia is myelogenous leukemia. It is the cancerous production of young myelogenous cells such as neutrophils, eosinophils, basophils, and monocytes. These young, immature white blood cells start out in the bone marrow and then spread throughout many parts of the body. The first effect of leukemia is a metastatic growth of leukemic cells in abnormal areas of the body. Leukemic cells from the bone marrow may reproduce so greatly that they invade the surrounding bone, causing pain and eventually a tendency for bones to fracture easily. In other words, the rate of production of these abnormal white blood cells becomes so great that the medullary cavity within the bone, expands to accommodate this rapidly dividing population of cells. Additionally, you need to appreciate that large numbers of these abnormal white blood cells are being released into the bloodstream. Although there are greater numbers of white blood cells, they are not functional white blood cells and do not confer immunity to invading microbes. Rather, they are abnormal and released in an immature state. So, although great in number, they are poorly differentiated and therefore useless. Additionally, the cancerous cells within the medullary cavity consume too many of nutrients, and this means that other parts of the body will suffer. Almost all types of cancer cause both anorexia, which means a reduction in food intake caused primarily by diminished appetite, and cachexia. This is a metabolic disorder of increased energy expenditure, leading to weight loss greater than that caused by food intake alone. Let me again stress the importance of non-specific host defenses. These constitute simple barriers and generic reactions to antigens and foreign microbes. The barriers include cutaneous and mucous membranes. The secretions from these membranes can prevent the spreading of invading microbes. Mucus can trap microbes. Serosal secretions may contain lysozymes or bactericides that can destroy the microbes. Then we have the phagocytic white blood cells that are generic in their responses to invading microbes, neutrophils, and tissue macrophages. Tissue macrophages are the first responders to an infection, and they are also considered to be antigen-presenting cells. Inflammation is also a non-specific host defense. The characteristics of inflammation include dolor, or condition of physical pain, mental anguish, or suffering from heat. It is one of the 4 signs of inflammation. The other 3 characteristics are calor, which means heat, rubor, which means redness, and tumor, which means swelling. Fever is another non-specific host defense, and this elevates metabolic rates for all cells of the body, including the activity of white blood cells. Lastly, antimicrobial substances are also non-specific. I would like to take the time right now to reiterate the specificity of T and B cells in their ability to react to only one antigen. Remember, an antigen is a polysaccharide, or protein, that can elicit an immune response. Not all parts of the antigen are necessarily recognized by the host's immune cells. Rather, only a particular portion of it, called an epitope, may be the antigenic region. Cytotoxic T cells and helper T cells express receptors on their cell surface that are capable of binding only one epitope of one particular antigen. The antibodies that are secreted by B cells can only recognize one epitope from a microbe. And when I think of this specificity, I think of a lock and key analogy. Perhaps you have used your house key to unlock your door. Perhaps you decided to do the empirical test and see if your house key was specific to your front door. You promptly walked to your neighbor's door and tried your key in their lock. Perhaps you discovered that your key, in fact, does not open your neighbor's door. Side note, in no way do I encourage you to perform this experiment, as it may cause you to experience extreme bodily harm or incarceration. My house key will only unlock my front door, not my neighbor's. Let's pretend that foreign antigens that you may encounter during your lifetime are represented as locks, and your T cells and B cells are keys. Each flu virus, bacterial infection, or fungal infection you may ever have in your lifetime is represented as a new lock entering your body. The T cells and B cells that fought the infection you had last month fit into that lock, but the infection you have this month is a new lock, and thus a new set of keys must be used to fight that infection. Obviously, this means we need millions of different types of B and T cells in our body. When one specific B or T cell encounters its antigen that it recognizes, it is activated and quickly creates copies of itself. Think of it as making duplicate keys. When the infection is over, many of the duplicated T or B cells die, while a few continue to live as, quote, memory cells. These memory cells will allow you to mount a quicker response and more aggressive response should you ever become infected with the same antigen in the future. It is now time to talk more about cell-mediated immunity. Again, this is a specific branch of acquired or specific immunity that arises through T cell action. There are three main T cells: cytotoxic T cells, helper T cells, and suppressor T cells. Suppressor T cell function is not well understood, but we do know they are capable of suppressing the functions of cytotoxic and helper T cells. They prevent the cytotoxic T cells from generating excessive immune reactions that could destroy healthy surrounding tissue. The helper T cell category is by far the most numerous. As their name implies, they help the immune system direct its response. They do this through several ways. In one way, they release many protein mediators called lymphokines. These are basically cytokines or chemicals that attract or stimulate other cells. These cytokines are called lymphokines because it is a lymphocyte that is making them, and because these proteins act on other immune cells as well as on bone marrow cells. I like to think of the helper T cell, also called a CD4 plus cell, as the Paul Revere of the white blood cells. Why do I think this? Well, a helper T cell interacts with a macrophage. Remember, it is an antigen-presenting cell. During this interaction, the helper T cell binds to the epitope that is presented by the macrophage on its cell surface. A helper T cell, like other T cells and B cells, is specific to recognize only one particular epitope from a foreign antigen. When the helper T cell is activated, it can then stimulate B cells to mature into plasma cells that can then secrete antibodies. I'll talk more about B cells in a bit. Shown on the far right is a cytotoxic T cell. The cytotoxic T cell must have direct contact with its target cell. It is capable of destroying microorganisms and may even destroy cells from the body. Basically, the cytotoxic T cell punches holes into the cell's membrane it is attacking, and the cell lyses. Now it's time to talk about the other branch of acquired immunity, and that is humoral immunity. This immunity is conferred by the B cells in your body. B cells provide protection against foreign antigens that are in your circulation or outside of a cell. These antigens may arise from bacteria, viruses, or parasites, just to name a few. When the B cell becomes activated by an antigen-presenting cell, such as the macrophage, or activated by a helper T cell, the B cell will mature into a plasma cell. The plasma cell can make antibodies. I'll talk about antibody structure in a little while. Again, I want to emphasize that the antibody will only be able to recognize one specific antigen. That means you would need many different types of B cells, each capable of making only one type of antibody to defend against the endless number of foreign antigens you may encounter. I'll go into antibody function in more detail later, but for right now it's sufficient to know that antibodies act to limit, inactivate, or aid in antigen destruction. And just like T cells, B cells create memory cells. This means if you ever encounter or become exposed to the same antigen at a later time, the memory cells can quickly create clones of themselves, and this means a quicker, more abundant supply of antibodies. Here is the basic structure of an antibody. This is a protein that is secreted by a mature B cell called a plasma cell. Antibodies are also called immunoglobulins. Antibodies are secreted into the plasma by the plasma cells. You'll notice that they are a Y-shaped protein with two long peptide chains that are colored here as dark brown, and two short chains that are colored here as red. The stem region determines the classification type of the antibody. There are 5 main classes. IG stands for immunoglobulin, and the classes are IgG, IgA, IgM, IgE, and IgD. I always remember this as gamed. The arm regions that project outward from the stem consist of amino acids that allow the antibody to bind to one particular antigen. There are usually only 2 variable regions, one at the end of each arm, and it is here that the antibody gets its specificity in binding one particular epitope on an antigen. Larger antibodies, such as IgMs, have multiple binding sites for a particular epitope. Here's a slide where I can stress to you the importance of understanding that B cells can only recognize one particular antigen. Shown at the top of the slide are 4 B cells with an integral protein shaped differently in each cell. You'll notice that only the one B cell that is square-shaped can recognize this square antigen. And once it does bind its antigen, that activates the B cell, and the B cell can actively replicate and divide and create more clones of itself. Additionally, that B cell will be stimulated into a plasma cell or a mature B cell. And it is the plasma cell that can now generate antibodies that will specifically bind to that square-shaped antigen. Again, these cells are specific to one particular antigen. This is similar to the T cells that can also respond to one particular antigen. Clearly, you would need many, many different B cells and T cells in order to fight any invading antigen or microbe that you might encounter throughout your lifespan. This concludes our discussion on Chapter 17, Blood Cell Types.